All right, I want to thank Sarah and her whole crew for this opportunity, and all of you for coming. I appreciate an audience and a chance to talk about the work that we do, trying to understand fossils better, and also trying to understand sort of the history of modern life better. A little about me. I'm a biologist at heart, and so I studied the fossil record from a biological perspective. I actually teach human anatomy to first-year medical students who are training to be doctors, but I combine that with an interest in dinosaurs uh, and how they move, and there's a lot more similarities there than you might expect. I'm going to let Morgan introduce herself um, when she does her part of this. We're going to kind of tag team it. I'll go first, and then we'll hand off, and hopefully that handoff will go smoothly. We'll see how that goes, and then you could ask questions of both of us or whatever you want to do at the end. As I said, my interest is in dinosaur locomotion, and so we're all familiar with Tyrannosaurus and these big sauropods and all these big giant extinct things. There certainly are a lot of smaller dinosaurs and dinosaurs when they were younger, but we're coming at these as living animals, right? They are fantastical, they are monstrous, they are um, things that are hard to imagine, and we're working to try to reanimate them in a biologically realistic way. Think about how they moved, and movement is pretty key to their whole evolutionary history, whether it's going from more sprawling forms to more erect forms, from 4 legs to 2 legs, um, evolving flight, changing body size. All these things are happening within this huge radiation of dinosaurs that were so successful. Like most paleontologists, we do study bones. And so if we're interested in how Tyrannosaurus walked or whether it could run or not, or how fast it could go, we're obviously going to take a look at that skeletal material, try to understand how the joints work, what kind of limits and possibilities those are imposing at each of those joints, how those joints might be coordinated. And we use a lot of studies of living animals in order to test our ideas on that. But I do want to emphasize we have to remember that skeletons don't really stand, even though they do in museums. They certainly don't walk. They can't really do anything by themselves. They're the framework on which animals have muscles, which produce forces and torques, and then their masses and inertias, and all those things are really what locomotion is about. And so skeletons are wonderful. They're critical. We would not know about the animals very well if we didn't have them, but there's a whole lot there missing. And to get to a truly locomoting creature, we need to try to reconstruct a lot of that missing information to try to understand how it worked. What's neat about tracks is it really comes at this locomotion question from a really different perspective and a really different angle. And so it is evidence of some kind of behavior, right? Our animal is interacting with its environment, interacting enough that it deforms the ground in a way that can get preserved. And then millions of years later, we can find that. Just, you really think about it, it's just pretty amazing that we're looking at this ground the dinosaur stepped on and distorted or bent or moved or in some way modified enough that we can then recognize it 200 million years later or whatever. Really fortunate for those of us working on locomotion because it is the behavior most of the time that we actually care about. If I was trying to study flight and how the wings move, or if I was interested in sound production and how the larynx moves or the syrinx or the lungs, those are going to be really hard to find direct evidence for that behavior simply because there's not that kind of permanent environmental deformation of the air. Whereas the ground, if those steps for the most part are what we're interested in. And so most of the tracks we find are locomotor, the animal walking or running or doing something with its legs, and with each step leaving behind evidence that we can try to make sense of. The other key here is that even though they're long dead, tracks were made while the animals were alive, right? So we're dealing with a biologically viable thing interacting with its environment, whereas the skeleton is evidence or preserves evidence of animal after it was dead. And so trying to reanimate that with evidence from the tracks then is really a nice complement. We can kind of work it from both sides. I don't want to say one is better, one is worse. There are times where you can get more from one than the other, but the more we can integrate them, the more we can combine them, I think the better off we are. That said, to really extract the information, particularly for a biologist like me, we want to make sure we're interpreting those tracks as correctly as we can. And sometimes it's not as straightforward as you might think, and that's one of the themes of the talk, is that footprints might seem quite simple, right? You step on the ground and you leave behind a mold of your foot. And that's true sometimes, but in many cases it's not. And trying to understand where variation in footprints come from is one of our lab themes. That'll be the theme of the talk today. I don't have a picture from my childhood. I wish I did, but this is far back enough that I had hair. And so this is me in 1989. I just finished grad school, kind of right where Morgan is in her stage of her training. And I went off to Greenland with a bunch of colleagues looking for skeletal material. And we found a lot of it, but we also found footprints. We were not specifically expecting footprints or looking for them specifically. These were just pretty much naturally exposed. I've swept them off with a broom. They're sitting here out on this cliff in Greenland. And once we started looking for them, they were everywhere. I'm not going to go into details of the Greenland work here, but just want to mention that it really was influential for my career, set me off in a different tangent that I hadn't expected, in part because they were these funny tracks that we found there. And so I've labeled the toes here with these Roman numerals. It's an animal that's putting the sole of its foot down. It's got 4 toe prints. It's got this big, funny looking Q-tip-like volcano at the front. And it's unusual because they seem to be walking around bipedally, just like a dinosaur would, with their feet kind of in line. But there's nothing out there from the late Triassic that has a foot that looks like this. And so this simple puzzle, and trying to solve this simple puzzle, really what led me to this very different view of what footprints are and what footprints could be. And we're going to try to share some of that with you today. Quick take home from Greenland is it's ultimately, footprints don't have to look like feet. I think we already know this at some level, but I didn't appreciate it. And so if the ground is soft enough and you sink deeply enough, you then move your foot through a motion that would normally happen in air, but that's now happening underground and preserving in a volume motion that we wouldn't normally be able to see. And that's really exciting for someone who works on the evolution of locomotion, to see evidence of locomotion, even if it's creating these funny tracks. So why birds? I think the most obvious reason is if we were looking for something around today that's most like a baby tyrannosaur, it would be our biggest flightless bird, something like this ostrich. Similar structure of the leg, obligate bipeds. They tend to stride rather than hop. They're standing up on their toes. There are obviously differences at the front end. It's very different from the ostrich. This big muscular tail is different in this guy. Lots and lots of subtle differences. And that's really where the fun begins, because we can say that a modern flightless bird like an ostrich is probably most similar to one of these extinct guys. But to say they're exact would take some real evidence. And so how are they different? How is this extinct animal unusual and unlike anything that's alive today, using evidence of what we have around us? If you saw Paul's talk, or you may already know, there's also an evolutionary reason why birds, and that is because if we look at a simplified dinosaur family tree, we find among living lizards and snakes, alligators and crocodiles and birds, what we think of as dinosaurs, these Mesozoic extinct dinosaurs, are most closely related to birds. And in fact, birds are dinosaurs. And so within this group of dinosaurs here, these guys are more related than they are to crocodiles, which are more related than they are to lizards and snakes. But we can really look at birds as this unusual branch of dinosaurs that evolved flight and then survived the extinction. So we have them around today. And if we didn't have them around today, I think we'd realize just how weird they are, right? We take them for granted. We eat them frequently. We live with them. We see them. We don't think about just how strange birds are. But these short-tailed, feathered, flying theropods are really kind of strange things. And so we want to take advantage of that, both because their limbs can be quite similar, particularly in these predatory theropod type forms, and because they're closely related. So we don't have to talk about a convergent evolution. In many cases, they are similarity by descent or shared descent. Lots of people doing track work, some of them trying to work with birds. Here's our colleague Jim Farlow walking along with his friendly emu here. And I say friendly because emus don't always have to be friendly. If you've ever dealt with large flightless birds, they can be dangerous. They can be hard to manage. They take a lot of space. They're expensive. They're very hard to control. And so you might think, well, while we're working on dinosaurs, we'd want the biggest bird we could find. Yes and no. And so I think not having to deal with this big bird has advantages, particularly if you're bringing them indoors. And so I got my start actually studying turkeys. And this is old video, grainy, sorry, back from the 90s of making this turkey or inducing this turkey to walk across this tray of mud to make tracks. Trying to get an early sense for what is the relationship between how this foot is moving and the track it leaves behind. Turkeys loose in a muddy room makes for a more muddy room, I can tell you. So we've gone even smaller since then. And most of the time we work with Guineafowl. You might be familiar with these guys. They're chicken-sized birds. Getting some feedback. They're from Africa. They love to walk and run rather than fly, although they can fly. If you take them into the lab, they'll actually run on a treadmill. You can train them to do that. And they're just kind of the right size for a lot of the equipment we have. And so turns out that some of the tracks we're studying aren't big giant tracks. They're actually smaller. And so Guineafowl work out just fine. Here's an example of a video we can take of a Guineafowl. And so now we've got foot interacting with a substrate in a living, breathing, real thing, right? We're not taking a fake foot and shoving it in the mud. It's deciding how far it sinks. It's deciding how it moves. It has to be viably locomoting. And so we think tracks have a lot of value. Can everyone else turn their mics off? Before we even get back to the lab, I want to just start with wild birds and some of the basic assumptions that people often make, and it's often reasonable assumptions. So these were out in the field behind my house a few winters ago. These are actually tracks on snow, which is maybe appropriate for what's happening outside. And if these were found in rock, paleontologists might start making up stories, right? This trail looks different than this trail looks. These two are maybe the same. Maybe they're different species of animals. Maybe they're all traveling together at the same time in a herd, who knows? A mixed species herd, how fast are they going? How tall were they? What were their bodies like? So we're making certain assumptions very, very quickly. Even linking these tracks together is an assumption, right? The fact that those make a trackway or a trail. In this case, the fact that they're different shapes makes perfect sense. So you may be familiar with these tracks. Wild turkey tracks in snow look very much like the bottom of a turkey foot in this case. And these other guys are actually webbed. And so those are our Canada goose tracks. And so anyone who's done hunting, anyone who's done tracking for fun, anyone who's just watched their pets or made tracks yourself at the beach or around any body of water or puddle, this relationship between foot shape and track shape is pretty tight, right? It's really our basis for being able to identify what creature is making that track. And if you look up a field guide of tracks, they're pretty much going to be tracks that look enough like the feet that you can identify them. Now imagine we find tracks like this in a rock, and imagine it's 1835, so prior to evolution, prior to dinosaurs. Edward Hitchcock's been mentioned a few times today. He was a professor and ultimately president of Amherst College back in the mid-1800s. And he was shown a slab of rock or two slabs of rock that had tracks like this on them, and immediately recognized that they were probably biological. They were probably made by animals walking. And he and others thought they looked an awful lot like bird tracks. He spent the next few months looking around at various quarries up and down the Connecticut River Valley from Hartford on up. Came up with other ones. I think Paul Olson just showed this big Eubrontes track, and did the first real publication on these things in 1836. And his wife drew out this beautiful diagram of these things. And so he said, hey, I've got big ones with fat toes, little ones with fat toes, big ones with skinny toes, some with 4 toes, some with 3 toes. Sometimes the 4th toe points forward, sometimes back. sometimes funny things going on in the heel. And just like our goose turkey example, he said, if I have different shaped tracks, I probably have different shaped feet, and those were probably made by different animals, therefore. And so he says, I've got 7 or so species back in 1836. Now, he continued to collect tracks and accumulate tracks and buy tracks and sort of grab tracks wherever he could over the next 30 years or so. And if you've been to the Benesky Museum of Natural History, where his collection is at Amherst College, you've experienced some of that collection, right? The walls and these tables are just covered with these giant slabs of rock covered in tracks, thousands of tracks. Those are the best known ones. What you might not be aware of is that under each of these tables are cabinets. Peter Falkingham, our close colleague on tracks, is opening up some of them. And within those, the white drawers, we then have thousands of more specimens, smaller specimens that are not typically shown. And so many of these might be something that could fit in the palm of your hand, right? It's a tiny little, maybe a single track, maybe multiple tracks, but they're small. They're kind of guineafowl size. They're not giant dinosaur tracks. They're little dinosaur tracks. And we've been trying to focus on understanding a lot of the diversity within these tiny little tracks. Because when Hitchcock continued to work, he just got, hey, I've got some like this. Oh, and I've got some like this. Some of these have narrow toes, some different angles, different lengths, different heel coverings, different hooks on the fourth toe, different length of the heel, different shape of the heel, sometimes parallel toes, sometimes curving, sometimes not. It just kept going and going and going, right? Describing everything he found. It was pretty amazing that he continued to do this. And when he got to his final major publication on this in 1858, his count was up to over 70. It all depends on how you count, of these animals that are walking either on 2 legs or had the potential to walk on 2 legs. And there were people who doubted that there were even 7 things alive, much less 70. And so the question comes, were there truly 70 different shaped feet out there making these different track types being possessed by 70 or more different species? It's possible. The other possibility is that he was kind of misinterpreting where these track shapes come from. And that's where we've been working. And so if you think about where track shape comes from, if you think it's entirely driven by the foot, then you have kind of a one-to-one relationship, right? The anatomy is what primarily dictates track shape. And that would fit with our turkey and goose example. But if you really think about how tracks are made, and people have done this, and Paul Olson and his colleague Kevin Padian have represented as this triangular diagram, it's not just anatomy that matters. The movement of the foot matters, and the consistency of the substrate matters. And so this kind of triangular push-pull of these various components interacting is really important. So the substrate can affect how you move, which then can affect the track shape. The anatomy can affect the substrate, which can affect track shape. And so the interplay between these are things we're trying to explore and see if we can explain Hitchcock's diversity, not just by 70 different kinds of feet, but by the formation process itself, involving substrate, motion, depth, things other than anatomy. And we're using birds for that. Here are some of the potential dinosaur track makers that I think people can agree on. There's probably some small sort of a ornithischian, a small theropod. I haven't shown the larger version of that, some kind of a prosauropod. And if we look in at the foot of our Coelophysis or Syntarsis, whatever we think it might be, we can see the similarity between its foot and our guineafowl foot that, again, Hitchcock was very aware of, and this is why he thought they were bird tracks. So 3 main toes pointing forward, a smaller 4th toe with variable orientations, but the same number of joints, the same number of segments, walking on their toes. Birds make sense for this. So let's see what happens if we start varying the substrate. We're going to start out firm. There's our guineafowl slowed down by 10 times, so we can really focus in on how that foot is planted. The other foot's going to swing through, plant in front of it, and then that rear foot is going to slowly roll up and off, just like we saw in that turkey video, only easier to see now. And if the substrate is firm enough that you don't sink too much, but not so firm that you don't leave a track, and has the right consistency, we can get a track that looks an awful lot like the mold of the bottom of the foot. Not exactly, These claw impressions still are digging in and are changing the shape of that claw. It's not an anatomical mold. It is still dynamic. But these toe impressions, 3 forward, 1 back, aren't just little depressions. They actually have details. And so you can see individual pads on the toes. You can see actual skin texture. We're getting down to the level of this is what that animal touched. And if we find that in the fossil record, we can make that same assumption. And so some of these tracks from the Benesky collection that have this pad detail and this claw detail, and some of them even have skin detail, we know we're dealing with similar firm substrates that are just right, not too sticky, not too dry, not too wet, not too slippery, where the animal can lay its foot down and give us a pretty decent approximation of what the anatomy, the soft tissue anatomy of that foot looked like. Those are not the ones we're going to spend our time on, because those are the easiest ones. And that's where most people spend their time, and rightly so, because that's the best place to get anatomy, the best place to identify the track maker. What if we put it on a sand-like substance, so dry grains? Turns out these are actually poppy seeds. Foot can't be supported anymore, so it plunges down and under, it gets covered up, and then has to re-emerge and sort of bursts its way back out again. And so obviously the tracks we're going to get from this substrate have to be very different from what we just saw in the firm mud, and they are. And I think I would challenge most people to say, are these guineafowl tracks? Are they bird tracks? What are they? There's just not a lot of detail there. We don't see pad impressions. We don't see claw impressions. It's a big collapsed, exploded thing, in part because those grains just won't hold any detail. We can also make the mud wetter. In this case, you plunge right on through. Depending on how wet we make it, the animal will sink to different degrees. It'll have mud all over its feet, but it cruises right on through this. But like those tracks from Greenland, we're now seeing, we're going to be capturing a lot of that motion. On the firm substrate, it will be happening above ground. On the soft mud, it's happening underground. And as a result, then the tracks do not look like feet. And this is our big theme here. If we were to really take a look at what's going on in one of these tracks, we can't just go and sort of label the toes like traditional track people would. We can't say, oh, there's 1 and 2 and 3 and 4. We can say this is where a toe went in, and this is where a toe went in, another toe went in. But this thing out in the front is not where the middle toe is. It's where the entire foot is withdrawn. And so the foot converges and is removed from the substrate, creating this big raised volcanic Q-tip thing, just like in the Greenland tracks that I got started on. And so again, this is an amazing kind of motion capture through a volume of substrate that for a locomotion person is really extremely valuable, but we have to be very careful then, because it doesn't look like a guineafowl foot anymore, or even a bird foot per se. So to take that home, here we have our guineafowl on firm mud, leaving foot-like tracks. A little bit wetter, we get things that you might think might even have webs as more of the foot engages. We go deeper still, they start looking like funny little star shapes. Deeper still, here's our pretty nondescript dry grains. And the wetter we go, the deeper you sink, the less like a foot you are, but the more motion you preserve. And so if you're interested in anatomy, you work at this end. If you're interested in motion, you kind of work at this end. But it should be humbling for anyone who thinks that they can take any track and tell me who made it, because these were all made not by just the same species, but actually the same bird. This is one individual that made all these. And so we think a lot of Hitchcock's diversity is simply substrate driven, that we're getting different shapes from the same foot, just the same way that if you walk on different parts of the beach, you can make different tracks. Dinosaurs can do that too. Here's some examples from dinosaurs, a shallow, firm, so firm that we're barely getting an impression from that middle toe, but we're seeing pads and claws. And here's some sloppier ones. Things start to drag, things start to collapse. They might not break very cleanly. I think these ones at the end here are the most guineafowl-like in terms of this big looping, sloppy thing. And you can imagine that the foot sunk quite a ways in. Maybe it's gone in up to its ankle or so, slogging its way through this. But it certainly doesn't look like the foot that made it anymore. It's really more about the motion and the flow of and collapse of the substrate. We are interested in variation with speed. It's a hard thing to get the animals to do. Here's a guineafowl on the ground running, but once it hits this stuff, they usually prefer not to keep going. We have no easy way to train them to go faster. But speed would be another potential variant. And so you might expect walking tracks to be different than running tracks. And we haven't really pursued much of that because we don't have very many of them. We take videos of our tracks from multiple perspectives if we can. We take lots of photos. In the old days, you would have to try to make a 3D model of this by pouring plaster in it or trying to potentially freeze it or in some way cast it or mold it. And then Peter Falkingham has been great using this modern technique where you take multiple photographs, maybe a few dozen photographs, and it's called photogrammetry. And from that, we can create a 3D digital model along with this photo texture to preserve that particular trial, which is nice. That means we can now smooth it over, run the bird again, and continue to do this work on the same sediment without having to try to maintain this. Now, as nice as that is to have the 3D model, we're only scratching the surface, right? We're looking at the air substrate boundary. We're saying, what did the animal see? It's the kind of way that we interact with tracks. You make a dent in the surface and you see it from above. But thinking like a geologist or thinking like a paleontologist, we have to realize there's stuff happening underneath there and that this is not the only surface that's getting disturbed. So if you heard some of the earlier talks on sedimentary rocks, either Paul's laminations or the earlier terrain lecture, this idea of laying down layers and layers of sediment, every time the water is bringing in sediment-laden material, covering things up and covering things up and covering things up. In this case, they've been tilted later on in life to get to those dinosaur tracks. Sometimes, like in Greenland, it'll naturally weather out, but that's pretty unusual. Most of the time, you're splitting rocks. You're going in with wedges, you're going in with pry bars, and then splitting this apart to then expose the tracks in between. And you need to think about what are the odds of that break happening right where the air was, the surface that the animal saw, the surface that was exposed at the time the track was made. It's just as likely, potentially more likely, that it could happen at a deeper layer or a shallower layer. And so we're not looking at that interface. And so we need to think about this more volumetrically, think about what's happening in deeper layers, not just the surface we're looking at. So we've done some very simple simulations here. I've taken a model of a dinosaur foot. We're going to simplify its middle toe down to a cylinder and then spin it, so we're looking on end, and then drive it down into this virtual substrate, which I've made like a little layer cake. And it's made up of these little particles. And so this is a simulation of a firm substrate creating what's going to end up being a shallow track. So here's our simplified toe model. We drive it down and we make a track. Now that's the idea here, right? This would be a track that you might be able to find. That one that the dinosaur touches would be called the true track. But look at all these disturbed layers underneath, here or here underneath the red one. Any of these breaks could potentially give you something that is a track. It is actually a track. It was made during the same event. The animal made it. It just didn't happen to touch it directly. And so we're calling these transmitted under tracks. These are some things we need to be aware of. And again, Hitchcock was aware of this, but it's a potential another source of variation. It gets more dramatic when the substrate gets sloppy. This will be our last slide. Here we're going to take our cylinder again, drive it into a much more hydrated mud. The animal doesn't stop, it keeps going. And not only does it keep going, but you can see that the walls of the trough it's making collapse and seal off. And so we basically don't really have an indentation anymore. We just have these deflected laminations from these different layers. And this is exactly what we find when we cut one of these fossil tracks with a rock saw. These downwardly deflected laminations from the original more horizontal sediment getting drawn down by a toe that would have passed through here. And if we break along one of these, and it could be anyone, it could be blue, it could be green, it could be yellow, orange, any of these surfaces can have the potential then to expose what we're calling a penetrative track. And so rather than looking like a toe, it's telling you someplace where the toe passed through. They tend to have these V-shaped contours very narrow toes, very sharp edge, don't have all that detail. And it's because they're made by a different mechanism. And so this penetrative track, we think, is the mechanism responsible for most, if not all, of the thin-toed track taxa that Hitchcock hypothesized. I'm going to leave it there and pass it off to Morgan.
Hello. Let me go on the slideshow.
You are there, Morgan.
Hi, everyone. Like Steve said, and as Sarah kindly introduced earlier, I am Morgan Turner. I am a grad student at Brown. I'm Steve's grad student. I'm actually graduating next week. And yes, I'm going to share a little bit more about kind of looking below the surface as the guineafowl makes these footprints, and kind of how that can help us interpret a lot of this track diversity that we're interested in trying to understand. And so, as Steve pointed out, this is our guineafowl on dry grains. We can't see the foot below the surface here. So we can see a few of those furrows on the surface, but the rest of the foot is obscured by the substrate. And here is kind of a smattering of diversity of these penetrative tracks that Steve was just describing to us. And I can highlight here some outlines in black of these different tracks. And we can see all sorts of different shapes happening, different numbers of seams in kind of where the toes passed, different exit points kind of indicated here by these open circles. And our fundamental question still remains, where does this track diversity come from? Is it from 70 plus species? Is it from substrate movement interactions? And as Steve showed before, this is our kind of triangular diagram of representing these different variables that can influence track shape. And of course, it's probably a combination of all three. And there we go. And so in the natural environment, animals encounter many different substrates, many things that you can imagine as you walk outside, like grass and leaf and snow. But ultimately, tracks that we're finding in the fossil record are typically preserved in these different muds and maybe sands. And so we're focusing our experiments here around replicating these substrates and really focusing in on the kind of interplay between the different substrates and the different motions that we get from our guineafowl. And so again, as Steve hit home earlier, avian and non-avian theropods share an incredible amount of similarities in their foot morphology or their foot shape, from the 3 anteriorly pointing toes that they walk primarily on, with an elevated metatarsus or tarsometatarsus, those fused long bones above. And so this bipedal animal interacts with the ground primarily through these 3 forward pointing toes. And as Steve kind of left us off with, tracks really are volumetric. The animal sees this upper surface and then plunges down below on any of these deformable surfaces or substrates. And so here again, I'm pointing out from the previous slide. And so this original surface is deformed in some way, and then we can't really see what's happening below the surface. We can't see exactly how the foot's moving. We can't see exactly how the substrate is deforming in response. And then eventually we see the foot exit out of that surface. And it's within that volume of substrate that is disturbed that potential fossil track slabs can come from. And so as Steve showed in that lamination, that where the rocks can be split and revealed at different depths, so can these different tracks. And if you can imagine, if you sink only 2 centimeters versus 8 centimeters, you're going to have a lot more potential tracks to sample from in a greater volume, or from, say, a wetter mud. And so this is something that we're exploring, is what is the foot doing below the ground? How is the foot interacting with these different substrates? And are there any patterns that can help us understand anything more about where this track diversity is coming from. And so to look underground to see how the foot is moving, we glued metal beads to the surface of the guineafowl foot here. And so we have the digits labeled 1 through 4, and we have some of these red markers here that are meant to represent our metal beads. And we used this method called X-ray reconstruction of moving morphology, or XROM, that was developed here at Brown by Steve and others. And it allows us to look underground and look inside bones. And we can see those metal beads, which are those black dots across the screen. And we can record with X-ray videography. And so we can have these movies of X-rays, and we can reconstruct in 3D the location, the positions of these metal markers through time and through space to actually capture where the foot has gone. And so to illustrate that, here's a video. So we can see the guineafowl walking in that dry grain, those poppy seeds. And here we can see below the surface, we're looking in the ground, and we can animate that foot based on those markers, and then trace the path of the tips of the claws here in blue from the entry paths, which are those thinner blue lines, and then the exit paths, which are those thicker ones. And so this is a 3D construct. These are 3D paths that we can sample. And of course, this isn't meant to represent the track, but this is where the foot went. And we can learn a lot from this, especially across all the different substrates that we tested. And so digit 3 here, so it's the longest digit, it sinks the lowest, the deepest consistently, and makes this looping pattern. If we look at the side toes, they don't always loop. They don't sink nearly as deeply. And then they converge as they exit altogether, as they pull out. And so these simple patterns we can explore a little bit more across the variation in the motion that we recorded, and across all the different substrates that we recorded the guineafowl walking on. And so here on the right, we have the surface of the ground represented by that black bar, and a path just tracing the tip of digit 3 here on the surface. And we can see that as the foot takes a step on that solid surface, the foot doesn't sink and doesn't slide. It just remains on the surface and then continues forward. But on all of these different substrates that we tested, the foot can sink deeply and create these looping patterns at different depths, never the same. And the foot can end up sinking past the ankle, which is, we're seeing the whole foot here. So the fused tarso-metatarsus, excuse my anatomical terminology, is visible here. And so the foot can sink quite deeply, but we see this consistent looping pattern from digit 3. And so what we can do is we can treat these loops and we can sample them like we were sampling these potential fossil slabs. So at different depths, we can cut it. And here is every 0.5 centimeter. And then what we can do is we can take those cuts, no matter where the loop is in space, and we can align them based on the entry placement. So where the foot originally sank. So we can kind of adjust them in this graph to the right. And while I know it's a lot to look at, you can see all of our diversity on the right with our few representative loops in color and then many others in gray. But you can pick up on a few very particular patterns. So one, at the very bottom, all these paths all approach 0. And so at some point at the very bottom of this loop, the foot turns around and lifts out. And the foot withdraws and pulls back as it's exiting. And so we end up getting a wider loop as we raise in the volume. And then eventually the digit 3 crosses its entry path and continues off in front. And so we have this consistent looping pattern of this digit that is really useful to keep an eye on when we're looking at depth in all these different fossils. Another major pattern that we've picked up on is, so here we're looking at an anterior view at the front of the foot, and we see that the toes are really spread as the foot sinks, and then as the foot withdraws, the toes collapse. And This is the same in figure form. And so we can see that at the bottom, the width of the side toes is quite a bit wider than higher up in the volume where they collapse. And again, this is another graph where we've kind of aligned all of our variable data on all these different substrates. But again, we see the same pattern occurring where the toes start off at the bottom of the loop very wide and then quickly converge. And so we can take all of that together and start to really understand the patterns that are happening in this volume, this track volume. And so near the top of the surface, I'm just going to kind of explain what this graphic is. So at the top of the surface, we'll have the bottom slab on the left. So these are our samples at depth. And then at the very bottom here, we have, say, at 13 centimeters down. And as we play through this animation on a really deep, wet mud, we can see these little kind of green-blue dots appear on these slabs down below. And that's where the claws actually pass through the slab. They don't have to pass at the same time, but that's where they encountered that depth. And as we continue on in the volume, or through this trial, we can see that at the end of this sinking phase, all the different claws end up at different depths, right? So digit 3 here at the very bottom is around 13 centimeters, and we can see that appearing down here at this bottom slab. But digit 4, say, over here only ends up going down past our 11 centimeter slab. And so we're getting a different sampling of these elements of anatomy here. And so as the foot withdraws, appearing kind of down at the bottom, we have these thicker blobs on our slabs, our virtual slabs. And we can see now that we have some separate exit locations. And we can continue further. And now we have a more converged toes. Sorry, I should use my pointers. And so our toes converge higher up in the volume, and we can see that also in the track slabs that these toes are more conjoined in space. And then lastly, from this very deep track, as the foot withdraws, it actually crosses out in front, much like that Greenland track Steve was showing us earlier. And so what we can do with this is we can prescribe zones based off these very simple features. If the toes are exiting out front from where they came into the surface, that is a zone 1 track, really high up in the volume. In the middle of the volume, when all of the toes are converged and they're exiting, but all of those exit features are behind the entry points, that is a zone 2 track. And then we get to the zone 3 tracks where we have separate exits separate exits, and they're all behind. And so for zone 3A, we have all 3 toes present. For zone 3B, we only have 2 toes present. And then for zone 3C, we have just the trace of digit 3. And so what we can do with that then is we can take this framework, this concept of how the foot is, this tridactyl foot that we know is anatomically very similar to many non-avian theropods and other dinosaurs. We think that for a variety of reasons from lots of research that has gone on in the past, that these bipedal dinosaurs move relatively similarly. And we can now apply this framework of guineafowl subsurface foot motion to, say, the Connecticut River Valley tracks that we have such a diversity of, and try to understand if there's another kind of dimension here that we can explore to understand the morphology that we're finding. And so here's one track. We can see we have 3 entry claw marks and 3 separate exit features. And so if we look back at our kind of representative guineafowl deep mud motion, we can get a sense that this track would be very low in the volume because we have those separate exit features, but we have all 3 digits present. And so that we could prescribe as zone 3A. We could flip this track over and look at the bottom and see that there's only a trace of digit 3. We have an entry and the exit here. And so we think we're even lower in the volume, which makes sense if this is the bottom of the slab. And this would be prescribed as zone 3C. And so here we can even see just from one slab that we're preserving somewhere really low in the track volume. Here's another track, also Connecticut River Valley. We can identify all 4 features from claws. We can also identify a single unified exit, which would suggest to us that it's zone 2. So it's a little bit kind of in the middle of of the volume that the foot stepped through. And there are some really awesome tracks, especially at the Benson Museum, these books where you can open up all of those different layers that were cracked open and see the sequence, the changes in the track shape with depth. And so this is one of those several surfaces that you can see. And the track shape changes with depth, and so do the entry and exit features. But most importantly, apologies, this is our kind of example of the track volume here, is that with depth, we also find this looping shape. And so the entry and the exit position of digit 3 in particular creates this looping shape that we found in Guineafowl, which is kind of further indicating to us that non-avian and avian dinosaurs are moving quite similarly, which is really cool to see. And so what this can do for us is we can identify entry and exit points on a diversity of fossil tracks. These are the same that I showed earlier. And there are hundreds. As Steve showed Peter kind of pulling through the drawers there, there are so many of these tracks. And we can now take a new approach to this diversity. And we can take these features and we can start to organize these shapes into zones. And so suddenly we go from not necessarily needing 70 plus species to make these track shapes, but rather just a very few are possible of making these. As we saw, the guineafowl can, just from sampling what the guineafowl can do, walking on a bunch of different muds, its movement is very different. And also how we sample that really matters. And so we propose really that there's this additional dimension of depth that's contributing substantially to track shape. And very much something important to consider when looking at and interpreting all of the motion that we see here. And so we hope that next time you visit a museum or see any of these penetrative tracks, well, hopefully that you can recognize them and understand just how much motion is being preserved in them. But it's not just the animal's motion, it's the motion and the flow of the substrate that the animal is walking on. And also consider where in the volume these tracks are being sampled from. We don't expect these feet to be identical to the tracks that we find. And it's really pretty fascinating to think about the amount of dynamic motion that happens the amount of dynamic motion that goes into creating one of these tracks and ultimately when we end up researching. So our research group, Steve, Peter, and I have been working at the Benson Museum. If you ever visit, hopefully in person soon. We also have the website here. We're working on putting together more of this information. Feel free to check it out. And thank you everyone for coming. We'll happily take some questions. Yes, thank you.
Thank you, Morgan. That was really, really interesting. I know I'm not visible. My voice is here, though. Can you access the Q&A from your computers, Steve and Morgan?
Yes.
There is a question in there.
I guess I should take that one. So Steve Winters asks, which substrate, laminated or uniform, is a better track maker? Which is more informative? Whatever I say, it'll become clear that I'm not a sedimentologist, Steve, and Paul's the person who knows this best. What I can tell you is that we've struggled we know we're never going to reconstruct the heterogeneous reality of the world. And so we haven't even tried for the most part. That firm mud I showed you was as uniform as we could make it. So we did not intentionally layer that. There are some times where we try to put a firm layer with a really soft layer on top of it, just to keep them from not sinking as far, but still have the fluid stuff on top. As far as we're concerned mechanically, since we don't really know how different laminae behave in terms of the rheology, we haven't found a need for lots of heterogeneity or laminations or bacterial mats or anything. We can create a huge diversity of natural looking tracks with relatively uniform substrates, or as uniform as we can make it. It's constantly changing. It's evaporating, the water's dropping out, it's doing various things. But I think Paul could tell you that probably the nicest fossil tracks come from certain places, and the crazy thin-toed leptodactylus ones come from other places, and we're still struggling to figure out exactly what those are. And it depends on you might better. I think they're all useful, and they're not necessarily the pretty ones, and we like the ones that aren't pretty. I hope that answered your question. You want to take the next one, Morgan?
Sure. Could sediment closure patterns indicate animal speed? That's a really interesting question. And speaking from a bit of experience just in kind of 3D printing some feet and plunging it into mud, I would say that there are several other factors that probably impact the closure. And I guess we haven't really explored a lot of speed, but if the foot is covered in mud and if it's really wet, that's going to really impact how the closure is happening, but also the consistency of the mud. And that might be really different in different levels of that substrate volume. And so I'm sure speed will have an effect, but I think ultimately that final closure, once the foot has left the ground, is really up to a lot of the properties that were kind of innate to the substrate before the animal even interacted with it.
Yeah, I'll add to that. Just what was interesting to me, these exit features, I think, are pretty much either ignored or just not understood in Hitchcock's time. And so he understood that the animals could sink through soft substrates substantially and that things would collapse. But as far as I can tell from the somewhat conflicting reading, he thought that the foot could get in and then back out, and then collapse would happen later. So you wouldn't necessarily be looking for an exit feature. Whereas what we're finding from our simulations and from physical modeling is that if it's soft enough to sink that deeply, you're pretty much collapsing as you go. That said, there are some Greenland tracks that maintain a slight gap, that stay slightly open, particularly for the metatarsus. But I don't know whether the impact of the speed, speed's hard to judge. I think that's the one we know very little about. Another one for you, Morgan.
Yes. What was my aha moment to choose to study dinosaur footprints? Well, when I applied to grad school, Steve had a dinosaur NSF grant and asked me if I was interested in working on them, and I said yes. And that's kind of ended up, I guess that was my first aha moment. I was kind of hooked from day one, analyzing a lot of this data. Yeah, I'll leave that there.
I'll say it was a slog, and trying to deal with the variation among all these patterns, every different substrate, every different step. What do you do with 80 or 100 different things that are not exactly comparable, because we don't know, they're not walking the same speed, the sediment's changing. And so Morgan was able to turn it around and sort of take advantage of the variation by finding that common loop pattern. I think that was probably the aha, which came later. But
I just put the website that I quickly had at the very end of my PowerPoint in the chat. Feel free to check it out. We will definitely be updating with more videos shortly.
Thank you.
Yes.
So Serdan, hi Serdan, we've been emailing, I think. Can you tell that a track is below the surface because the texture of the rock is smooth rather than having marks like ripples and raindrops if it was on the surface? We're struggling with that. There is definitely a particular smooth roundness to the deeper penetrative tracks, the way they collapse, and sometimes the true track at the true surface might be more angular. But mud cracks can travel down afterwards, right, and go deeper than potentially affect a deeper track. So just saying it's cracked meant it had to dry out. We don't know when it dried out, or it could have rippled and then been re-wet. And so I think we get sequences of things, and this confused Hitchcock because sometimes you find shallow, clear, anatomically more reliable tracks next to a penetrative track on the same slab. And that was part of his justification for saying that they were real creatures, that they were thin-toed things and thick-toed things. But I can imagine a variety of scenarios where it's firm at one point, animals make shallow tracks, you then pour more sediment on top of it, rehydrate it, and then penetrate it from above. And so the exact contemporaneous nature of any one surface is really, gets fun, gets interesting. Paul asked for the website.
Yes.
I'll just say we haven't done, we did a very preliminary thing with some thin mud with Morgan's alligators on them, but it really wasn't about the tracks, it was more about the bones. I'm involved with a physical anthropologist who studies hominid tracks from East Africa, and we're actually studying how human footprints are made. And so we're putting 85 or so little metal markers on the bottom of people's feet and seeing how their foot deforms underground and how their motion differs from the final track. And so it's the same concept. We don't get quite as much variation in humans. Most human tracks still look like human feet. And so it's much more subtle differences in arch height and heel height and ball shape. What's so much fun about the stuff that Morgan talked about is that these very narrow toes, so much of the track is getting in and getting out, and that they really can create a huge amount of diversity, even step to step in the same substrate.
Yes. Do you want to take the sauropod questions?
Go for it. We need Peter.
Yeah, we need Peter. Our collaborator studies sauropods. So,
can you read it? I'm not sure.
Yes, I can read it. Apologies. So Johan, 5 years old from India, asked, for sauropods, since they were heavier, how deep would the footprints go in wet mud? I think it's probably possible they can go quite deep. I'm sure they bottom out at kind of different depths depending on how deep the water can reach and all these other things. But ultimately, kind of like humans, sauropods have a really big flat foot. And that's very different from these very narrow, toes where there's a lot of space between them. And so I think it's really hard to say, and it's hard to compare animals with such different masses and how deep they would sink comparatively. I'm sure because they were very heavy, they could go quite deep.
But there are definitely tracks, Johan, from Australia I can think of, and other places, even the Western US, where the sauropods are sinking in 1, 2, 3 feet even. You can just see this giant pothole that they've created and taken down all those layers with them. But because it is kind of like an elephant foot, it doesn't really get swamped and covered over the way these narrow-toed guys do. Good question.
Yes.
Serdan asks again. Or you want to go?
No, you had.
Could soft substrate tracks be used to estimate movement speed if you were basing the estimation off of foot length and stride length, or would you not be able to use those tracks because they don't preserve the shape of the foot? That's another great question. So when I first got started in Greenland, I was interested in, my thought was if you encountered really soft stuff, maybe you slow down, that you would sense that. And that was actually our first idea. We thought that these guys with narrow toes and skinny legs didn't want to sink up to their knees and get stranded. so that maybe they were dropping down into a more flat-footed posture. So we were trying to use the relationship that Alexander and others have established between a step length and a hip height and a speed, but what length do you use when you get a track that you know is extra long? So I tried it with width, I tried it with some ratio, I tried it with a bunch of things. And I didn't see any good evidence that they were slowing down. As we've worked our way along, and Morgan mentioned these models, even the width I don't trust, because I think the substrate can flow and make the foot look narrower, even as it's making it look longer. So speed's a tough one there, but they don't, in Greenland anyway, where we have big exposures, they don't shy away from it. It's not like they say, it's getting too wet and turn right around. They'll go cruising right on through it 20 steps in a row. And I don't think they were struggling. I don't think they're suffering. They just kind of walk right on through it.
Scott asks, what's new and coming in technology to further this research, i.e. VR or AI? Steve might have a few slides up on some more of the particle simulation. Because this is such a volumetric phenomenon, we have collaborated for many years now with some virtual reality experts and computer scientists here at Brown, and we've been kind of exploring this very volumetric thing at the particle level, at the surface level, and within the volume, and tracing a lot of these trajectories. Are you going to?
Yeah, I'm trying.
There.
So this is something we did a few years ago. Morgan showed it on poppy seeds. And so we're putting in the whole limb skeleton here based on the 2 X-ray systems. And then Peter Falkingham is running a particle simulation rather than just a little cylinder cutting through layers. We can animate those cylinders into a foot and try to see if we can reconstruct what's happening on poppy seeds. And so on the left here, this is what we get at the surface, and it's just going to end up being that sort of nondescript track that isn't very informative. But if we virtually dig down a centimeter, we can see that even though these are dry grains, there's no cohesion. They fall like dry sand. We can get really interesting sharp contours because those grains are supporting each other. There's kind of nowhere to go. And so we can get sharp edges, steep features that you'd never see at the surface, just slightly below ground. Sorry, I'm trying to advance and it's not letting me. So this is one idea we had, was to really think about where the track is coming from, both at different depths and through time. And so this idea that if it starts out green, blue colors mean you're being pushed down, red colors mean you're being pulled up. And so we can see here the bottom of the foot sort of descending, starting to reach these deeper layers. And then as you come back out again, you're creating these big red features. And ultimately, this is the only one we get to see, right? The only final one. Because these other ones are transient in time and these guys are covered up. But all these are potential fossil surfaces. And so it's a little bit humbling to think that you might find something down here and say, oh, it's just a shallow track on firm mud, when in fact you're 3 centimeters down in something much more compliant. I think Morgan showed this one fossil, which does have these features. And I don't, I looked at this thing for years and I had ignored it. People just didn't really talk about it. But if we say, what's going on with that thing, right? Is it a mutation? Is this really is it a tumor? Is it a flange off the toe? If we actually go and look at what we have from our guineafowl, those are where the toes are coming back out again. So the side toes have come in, the back toe has come front, the middle toe has come back, and they're creating these little tubes as the sediment is drawn up and out. And that's what we're seeing here. There's actually a 3-sided structure representing the exit of that toe. That now that we know to look for exits, we look at tracks very, very differently. And I think that's the theme, is that we have very different eyes than we had even 5 years ago. This is one of these simulations that Peter's been doing. And so we're trying to think of what, if we're a layer somewhere deep in the volume, what might be happening to give us some of this track diversity. So we're putting in a relatively normal looking foot and coming up with a track that looks quite different. So everything shifts. I'll stop there.